genetically modified foods are foods produced from organisms that have had specific changes introduced into their DNA using the methods of genetic engineering. These techniques have allowed for the introduction of new traits as well as a far greater control over a food's genetic structure than previously afforded by methods such as selective breeding and mutation breeding. Commercial sale of genetically modified crops began in 1994, when Calgene first marketed its Flavr Savr delayed ripening tomato. To date, most genetic modification of foods have primarily focused on cash crops in high demand by farmers such as soybean, corn, canola, and cottonseed oil. These have been engineered for resistance to pathogens and herbicides and better nutrient profiles. GM livestock have also been experimentally developed, although as of November 2013 none are currently on the market. There is broad scientific consensus that food on the market derived from GM crops poses no greater risk to human health than conventional food. However, opponents have objected to GM foods on several grounds, including safety issues, environmental concerns, and economic concerns raised by the fact that GM seeds that are food sources are subject to intellectual property rights owned by multinational corporations. History: Food biotechnology is a branch of food science in which modern biotechnological techniques are applied to improve food production or food itself. Different biotechnological processes used to create and improve new food and beverage products include industrial fermentation, plant cultures, and genetic engineering. The use of food biotechnology dates back to thousands of years ago to the time of the Sumerians and Babylonians. These groups of people used yeast to make fermented beverages such as beer. The use of plant enzymes such as malts were also used millennia ago, before there was even an understanding of enzymes. Further advancement in food biotechnology occurred with the invention of the microscope by Anton van Leeuwenhoek, which allowed for humans to discover microorganisms which would then be used in food production. Food biotechnology was advanced in 1871 when Louis Pasteur discovered that heating juices to a certain temperature would kill off bad bacteria which would affect wine in fermentation. This process was then applied to milk production, heating milk to a certain temperature to improve food hygiene. Food science and food biotechnology was then progressed to include the discovery of enzymes and their role in fermentation and digestion of foods. With this discovery, further technological development of enzymes emerged. Typical industrial enzymes used plant and animal extracts, but this was later substituted by microbial enzymes. An example of this would be the use of chymosin in the production of cheese. Cheese was typically made using the enzyme Rennet, which would be extracted from the stomach lining of the cow. Scientists then started using a recombinant chymosin in order for milk clotting, resulting in cheese curds. Food enzyme production using microbial enzymes was the first application of genetically modified organisms in food production. Food biotechnology has grown to include cloning of plants and animals, as well as more development in genetically modified foods in more recent years. Scientists first discovered that DNA can transfer between organisms in 1946. The first genetically modified plant was produced in 1983, using an antibiotic-resistant tobacco plant. In 1994, the transgenic Flavr Savr tomato was approved by the FDA for marketing in the U.S. The modification allowed the tomato to delay ripening after picking. In the early 1990s, recombinant chymosin was approved for use in several countries, replacing rennet in cheese making. In the U.S. in 1995, the following transgenic crops received marketing approval: canola with modified oil composition, Bacillus thuringiensis corn maize, cotton resistant to the herbicide bromoxynil, Bt cotton, Bt potatoes, soybeans resistant to the herbicide glyphosate, virus-resistant squash, and additional delayed ripening tomatoes. In 2000, with the creation of golden rice, scientists genetically modified food to increase its nutrient value for the first time. As of 2011, the U.S. leads a list of multiple countries in the production of GM crops, and 25 grams crops had received regulatory approval to be grown commercially. As of 2013, roughly 85% of corn, 91% of soybeans, and 88% of cotton produced in the United States are genetically modified. Method of production. Genetically engineered plants are generated in a laboratory by altering their genetic makeup and are tested in the laboratory for desired qualities. This is usually done by adding one or more genes to a plant's genome using genetic engineering techniques. Most genetically modified plants can be modified in a directed way by gene addition or gene subtraction. Plants are now engineered for insect resistance, fungal resistance, viral resistance, herbicide resistance, changed nutritional content, improved taste, and improved storage. Once satisfactory plants are produced, sufficient seeds are gathered, and the companies producing the seed need to apply for regulatory approval to field test the seeds. If these field tests are successful, the company must seek regulatory approval for the crop to be marketed. Once that approval is obtained, the seeds are mass-produced and sold to farmers. The farmers produce genetically modified crops, which also contain the inserted gene and its protein product. The farmers then sell their crops as commodities into the food supply market, in countries where such sales are permitted. Foods with protein or DNA remaining from GMOs. As of 2013 there are several GM crops that are food sources and there are no genetically modified animals used for food production. In some cases, the plant product is directly consumed as food, but in most cases, crops that have been genetically modified are sold as commodities, which are further processed into food ingredients. Fruits and vegetables. Papaya has been genetically modified to resist the ring spot virus. Sunup is a transgenic red-fleshed sunset cultivar that is homozygous for the coat protein gene of PRSV. Rainbow is a yellow-fleshed F1 hybrid developed by crossing Sunup and non-transgenic yellow-fleshed Cappaho. The New York Times stated that in the early 1990s, Hawaii unregistered trademark S papaya industry was facing disaster because of the deadly papaya ring spot virus. Its single-handed savior was a breed engineered to be resistant to the virus. Without it, the state unregistered trademark S papaya industry would have collapsed. Today, 80% of Hawaiian papaya is genetically engineered, and there is still no conventional or organic method to control ring spot virus. The new leaf potato, brought to market by Monsanto in the late 1990s, was developed for the fast food market, but was withdrawn from the market in 2001 after fast food retailers did not pick it up and food processors ran into export problems. There are currently no transgenic potatoes marketed for human consumption. In October 2011 BASF requested cultivation and marketing approval as a feed and food from the EFSA for its Fortuna potato, which was made resistant to late blight by adding two resistance genes, BLB1 and BLB2, which originate from the Mexican wild potato Solanum bulbocastanum. However, in February 2013 BASF withdrew its application. In May 2013, the J.R. Simplot company sought USDA approval for their innate potatoes, which contain 10 genetic modifications that prevent bruising and produce less acrylamide when fried than conventional potatoes. The inserted genetic material comes from cultivated or wild potatoes, and leads to RNA interference, which prevents certain proteins from being formed. As of 2005, about 13% of the zucchini grown in the U.S. was genetically modified to resist 3 viruses. The zucchini is also grown in Canada. As of 2012, an apple that has been genetically modified to resist browning, known as the non-browning Arctic apple produced by Okanagan Specialty Fruits, was awaiting regulatory approval in the U.S. and Canada. A gene in the fruit has been modified such that the apple produces less polyphenol oxidase, a chemical that manifests the browning. Milled corn products. Corn used for food has been genetically modified to be resistant to various herbicides and to express a protein from Bacillus thuringiensis that kills certain insects. About 90% of the corn grown in the U.S. has been genetically modified. Human-grade corn can be processed into grits, meal, and flour. Grits are the coarsest product from the corn dry milling process. Grits vary in texture and are generally used in cornflakes, breakfast cereals, and snack foods. Brewers unregistered trademark grits are used in the beer manufacturing process. Cornmeal is an ingredient in several products including cornbread, muffins, fritters, cereals, bakery mixes, pancake mixes, and snacks. The finest grade cornmeal is often used to coat English muffins and pizzas. Cornmeal is also sold as a packaged good. Corn flour is one of the finest textured corn products generated in the dry milling process. Some of the products containing corn flour include mixes for pancakes, muffins, donuts, breadings, and batters, as well as baby foods, meat products, cereals, and some fermented products. Masa flour is another finely textured corn product. It is produced using the alkaline cooked process. A related product, masa dough, can be made using corn flour and water. Masa flour and masa dough are used in the production of taco shells, corn chips, and tortillas. Milled soy products. About 90% of the planted area of soybeans in the U.S. are genetically modified varieties. Soybean seeds contain about 20% oil. To extract soybean oil from the seeds, the soybeans are cracked, adjusted for moisture content, rolled into flakes and solvent extracted with commercial hexane. The remaining soybean meal has a 50% soy protein content. The meal is toasted and ground in a hammer mill. 98% of the U.S. soybean crop is used for livestock feed. Part of the remaining 2% of soybean meal is processed further into high-protein soy products that are used in a variety of foods, such as salad dressings, soups, meat analogues, beverage powders, cheeses, non-dairy creamer, frozen desserts, whipped topping, infant formulas, breads, breakfast cereals, pastas, and pet foods. Processed soy protein appears in foods mainly in three forms: soy flour, soy protein isolates, and soy protein concentrates. Soy protein isolates. Food-grade soy protein isolate first became available on October 2, 1959 with the dedication of Central Soy's edible soy isolate, Promine D, production facility on the Glidden Company industrial site in Chicago. Soy protein isolate is a highly refined or purified form of soy protein with a minimum protein content of 90% on a moisture-free basis. It is made from soybean meal which has had most of the non-protein components, fats and carbohydrates removed. Soy isolates are mainly used to improve the texture of processed meat products, but are also used to increase protein content, to enhance moisture retention, and are used as an emulsifier. Soy protein concentrates. Soy protein concentrate is about 70% soy protein and is basically soybean meal without the water-soluble carbohydrates. Soy protein concentrate retains most of the fiber of the original soybean. It is widely used as a functional or nutritional ingredient in a wide variety of food products, mainly in baked foods, breakfast cereals, and in some meat products. Soy protein concentrate is used in meat and poultry products to increase water and fat retention and to improve nutritional values. Flours. Soy flour is made by grinding soybeans into a fine powder. It comes in three forms: natural or full fat, defatted with 50% protein content and with either high water solubility or low water solubility, and lessifinated. As soy flour is gluten-free, yeast-raised breads made with soy flour are dense in texture. Soy grits are similar to soy flour except the soybeans have been toasted and cracked into coarse pieces. Kinoko is a soy flour used in Japanese cuisine. Textured soy protein. Textured soy protein is made by forming a dough from soybean meal with water in a screw-type extruder, and heating with or without steam. The dough is extruded through a die into various possible shapes and dried in an oven. The extrusion technology changes the structure of the soy protein, resulting in a fibrous, spongy matrix similar in texture to meat. Teaspoon is used as a low-cost substitute in meat and poultry products. Highly processed derivatives containing little to no DNA or protein, lecithin. Corn oil and soy oil, already free of protein and DNA, are sources of lecithin, which is widely used in processed food as an emulsifier. Lecithin is highly processed. Therefore, GM protein or DNA from the original GM crop from which it is derived is often undetectable with standard testing practices. In other words, it is not substantially different from lecithin derived from non-GM crops. Nonetheless, consumer concerns about genetically modified food have extended to highly purified derivatives from GM food, like lecithin. This concern led to policy and regulatory changes in Europe in 2000, when Regulation 50/2000 was passed, which required labeling of food containing additives derived from GMOs, including lecithin. Because it is nearly impossible to detect the origin of derivatives like lecithin with current testing practices, the European regulations require those who wish to sell lecithin in Europe to use a meticulous system of identity preservation. Vegetable oil. Most vegetable oil used in the U.S. is produced from several crops, including the GM crops canola, corn, cotton, and soybeans. Vegetable oil is sold directly to consumers as cooking oil, shortening, and margarine, and is used in prepared foods. There is no, or a vanishingly small amount of, protein or DNA from the original GM crop in vegetable oil. Vegetable oil is made of triglycerides extracted from plants or seeds and then refined, and may be further processed via hydrogenation to turn liquid oils into solids. The refining process removes all, or nearly all non-triglyceride ingredients. Cornstarch and starch sugars, including syrups. Starch or amylum is a carbohydrate consisting of a large number of glucose units joined by glycosidic bonds. This polysaccharide is produced by all green plants as an energy store. Pure starch is a white, tasteless and odorless powder that is insoluble in cold water or alcohol. It consists of two types of molecules, the linear and helical amylose and the branched amylopectin. Depending on the plant, starch generally contains 20 to 25% amylose and 75 to 80% amylopectin by weight. To make cornstarch, corn is steeped for 30 to 48 hours, which ferments it slightly. The germ is separated from the endosperm and those two components are ground separately. Next, the starch is removed from each by washing. The starch is separated from the corn steep liquor, the cereal germ, the fibers and the corn gluten mostly in hydrocyclones and centrifuges, and then dried. This process is called wet milling and results in pure starch. The products of that pure starch contain no GM DNA or protein. Starch can be further modified to create modified starch for specific purposes, including creation of many of the sugars in processed foods. They include maltodextrin, a lightly hydrolyzed starch product used as a bland-tasting filler and thickener. Various glucose syrups, also called corn syrups in the U.S., viscous solutions used as sweeteners and thickeners in many kinds of processed foods. Dextrose, commercial glucose, prepared by the complete hydrolysis of starch. High fructose syrup, made by treating dextrose solutions with the enzyme glucose isomerase, until a substantial fraction of the glucose has been converted to fructose. In the United States, high fructose corn syrup is the principal sweetener used in sweetened beverages because fructose has better handling characteristics, such as microbiological stability, and more consistent sweetness flavor. One kind of high fructose corn syrup, HFCS 55, is typically sweeter than regular sucrose because it is made with more fructose, while the sweetness of HFCS 42 is on par with sucrose. Sugar alcohols, such as maltitol, erythritol, sorbitol, mannitol and hydrogenated starch hydrolysate, are sweeteners made by reducing sugars. Sugar. The United States imports 10% of its sugar from other countries, while the remaining 90% is extracted from domestically grown sugar beet and sugarcane. Of the domestically grown sugar crops, half of the extracted sugar is derived from sugar beet, and the other half is from sugarcane. After deregulation in 2005, glyphosate-resistant sugar beet was extensively adopted in the United States. 95% of sugar beet acres in the U.S. were planted with glyphosate-resistant seed in 2011. Sugar beets that are herbicide-tolerant have been approved in Australia, Canada, Colombia, EU, Japan, Korea, Mexico, New Zealand, Philippines, Russian Federation, Singapore, and USA. The food products of sugar beets are refined sugar and molasses. Pulp remaining from the refining process is used as animal feed. The sugar produced from GM soya beets is highly refined and contains no DNA or protein. It is just sucrose, the same as sugar produced from non-GM soya beets. Foods processed using genetically engineered products. Cheese. Rennet is a mixture of enzymes used to coagulate cheese. Originally it was available only from the fourth stomach of calves, and was scarce and expensive, or was available from microbial sources, which often suffered from bad tastes. With the development of genetic engineering, it became possible to extract rennet-producing genes from animal stomach and insert them into certain bacteria, fungi or yeasts to make them produce chymosin, the key enzyme in rennet. The genetically modified microorganism is killed after fermentation and chymosin isolated from the fermentation broth, so that the fermentation-produced chymosin used by cheese producers is identical in amino acid sequence to the animal source. The majority of the applied chymosin is retained in the whey and some may remain in cheese in trace quantities and in ripe cheese. The type and provenance of chymosin used in production cannot be determined. FPC was the first artificially produced enzyme to be registered and allowed by the U.S. Food and Drug Administration. FPC products have been on the market since 1990 and have been considered in the last 20 years the ideal milk clotting enzyme. In 1999, about 60% of U.S. hard cheese was made with FPC and it has up to 80% of the global market share for Rennet. By 2008, approximately 80% to 90% of commercially made cheeses in the US and Britain were made using FPC. Today, the most widely used fermentation-produced chymosin is produced either by the fungus Aspergillus niger and commercialized under the trademark CHY marks, a registered trademark by the Danish company CHR Hansen, or produced by Cluiveromyces lactis and commercialized under the trademark Maxa and a registered trademark by the Dutch company DSM. Foods made from animals fed with GM crops or treated with bovine growth hormone, livestock and poultry are raised on animal feed, much of which is composed of the leftovers from processing crops, including GM crops. For example, approximately 43% of a canola seed is oil. What remains is a canola meal that is used as an ingredient in animal feed and contains protein from the canola. Likewise, the bulk of the soybean crop is grown for oil production and soy meal, with the high-protein defatted and toasted soy meal used as livestock feed and dog food. 98% of the U.S. soybean crop is used for livestock feed. As for corn, in 2011, 49% of the total maize harvest was used for livestock feed. Despite methods that are becoming more and more sensitive, tests have not yet been able to establish a difference in the meat, milk, or eggs of animals depending on the type of feed they are fed. It is impossible to tell if an animal was fed GM soy just by looking at the resulting meat, dairy, or egg products. The only way to verify the presence of GMOs in animal feed is to analyze the origin of the feed itself. In some countries, recombinant bovine somatotropin is approved for administration to dairy cows in order to increase milk production. RBST may be present in milk from RBST-treated cows, but it is destroyed in the digestive system and even if directly injected, has no direct effect on humans. The Food and Drug Administration, World Health Organization, American Medical Association, American Dietetic Association, and the National Institute of Health have independently stated that dairy products and meat from BST-treated cows are safe for human consumption. However, on September 30, 2010, the United States Court of Appeals, Sixth Circuit, analyzing evidence submitted in briefs, found that there is a compositional difference between milk from BGH-treated cows and milk from untreated cows. The court stated that milk from BGH-treated cows has increased levels of the hormone insulin like growth factor 1, higher fat content and lower protein content when produced at certain points in the cow's lactation cycle, and more somatic cell counts, which may make the milk turn sour more quickly. Foods made from GM animals. As of November 2013 there were no genetically modified animals approved for use as food, but a gram salmon was awaiting regulatory approval at that time. Animals usually used for food production have already been genetically modified and approved by the FDA and EMA to produce non-food products. One of the biggest obstacles for GM animals to enter the food market is the social acceptance of it. There is currently a huge debate as the first GM animal, salmon, is approaching commercial market. The possibility of modifying other animals as food has also been discussed but not yet underway. Research and experiments have gone into adding promoter genes into animals to increase growth speed, and increasing resistance of disease. Controversies. The genetically modified foods controversy is a dispute over the use of food and other goods derived from genetically modified crops instead of from conventional crops, and other uses of genetic engineering in food production. The dispute involves consumers, farmers, biotechnology companies, governmental regulators, non-governmental organizations, and scientists. The key areas of controversy related to GMO food are whether GM food should be labeled, the role of government regulators, the objectivity of scientific research and publication, the effect of GM crops on health and the environment, the effect on pesticide resistance, the impact of GM crops for farmers, and the role of GM crops in feeding the world population. There is broad scientific consensus that food on the market derived from GM crops poses no greater risk than conventional food. No reports of ill effects have been documented in the human population from GM food. The starting point for assessing the safety of all GM food is to evaluate its substantial equivalence to the non-modified version. Further testing is then done on a case-by-case basis to ensure that concerns over potential toxicity and allergenicity are addressed prior to a GM food being marketed. Although labeling of genetically modified organism products in the marketplace is required in 64 countries, in the United States, there is no general requirement that GMO foods must be labeled as such. The FDA's policy is to require a specific label if there are significant differences in composition or differences that are material to health, but it has not found any such differences in any GMO food currently approved for sale. Opponents of genetically modified foods such as the advocacy groups Organic Consumers Association, the Union of Concerned Scientists, and Greenpeace claim risks have not been adequately identified and managed, and they have questioned the objectivity of regulatory authorities. Some health groups say there are unanswered questions regarding the potential long-term impact on human health from food derived from GMOs, and propose mandatory labeling or a moratorium on such products. Concerns include contamination of the non-genetically modified food supply, effects of GMOs on the environment and nature, the rigor of the regulatory process, and consolidation of control of the food supply and companies that make and sell GMOs. Regulation. Governments have taken different approaches to assess and manage the risks associated with the use of genetic engineering technology and the development and release of genetically modified organisms, including genetically modified crops and genetically modified fish. There are differences in the regulation of GMOs between countries, with some of the most marked differences occurring between the USA and Europe. Regulation varies in a given country depending on the intended use of the products of the genetic engineering. For example, a crop not intended for food use is generally not reviewed by authorities responsible for food safety. One of the key issues concerning regulators is whether GM products should be labeled. Labeling can be mandatory up to a threshold GM content level or voluntary. A study investigating voluntary labeling in South Africa found that 31% of products labeled as GMO-free had a gram content above 1.0%. In Canada and the USA labeling of GM food is voluntary, while in Europe all food or feed which contains greater than 0.9% of approved GMOs must be labeled. As of 2013, 64 countries require GMO labeling. More than a third of these under a single EU ruling. Detection. Testing on GMOs in food and feed is routinely done using molecular techniques like DNA microarrays or quantitative PCR. These tests can be based on screening genetic elements or event-specific markers for the official GMOs. The array-based method combines multiplex PCR and array technology to screen samples for different potential GMOs, combining different approaches. The qPCR is used to detect specific GMO events by usage of specific primers for screening elements or event-specific markers. Controls are necessary to avoid false positive or false negative results. For example, a test for CAMV is used to avoid a false positive in the event of a virus-contaminated sample. In a January 2010 paper, the extraction and detection of DNA along a complete industrial soybean oil processing chain was described to monitor the presence of Roundup-ready soybean. The amplification of soybean lectin gene by endpoint polymerase chain reaction was successfully achieved in all the steps of extraction and refining processes, until the fully refined soybean oil. The amplification of a soybean by PCR assays using event-specific primers was also achieved for all the extraction and refining steps, except for the intermediate steps of refining, possibly due to sample instability. The real-time PCR assays using specific probes confirmed all the results and proved that it is possible to detect and quantify genetically modified organisms in the fully refined soybean oil. To our knowledge, this has never been reported before and represents an important accomplishment regarding the traceability of genetically modified organisms in refined oils. See also, California Proposition 37, chemophobia, genetic engineering, genetically modified crops, genetically modified food controversies, genetically modified organisms, regulation of the release of genetic modified organisms, Starlink corn recall, references. External links, documentary on YouTube, library resources in your library and in other libraries about genetically modified food, media related to genetically modified organisms at Wikimedia Commons.